Have you ever wondered why you can perfectly remember a random moment from when you were 3 years old, but you can't remember where you put your keys 5 minutes ago? Well, scientists just discovered something incredible about how our brains create memories that can literally last a lifetime. And get this, it all started with a neuroscientist trying to understand his only memory of his sister who died when he was just a toddler. Picture this. Todd Sacktor was about to turn 3 when his 4-year-old sister died of leukemia. An empty bedroom next to his, a swing set with 2 seats instead of 1, and just one single memory that survived over 60 years. His sister brushing him off when he asked her to read him a book, telling him to go ask his mother instead. That's it. One fleeting moment, but it stuck with him for 6 decades. Now here's where it gets wild. That memory isn't just some abstract thought floating around in his head. It's literally a physical trace etched into his brain tissue by molecular machinery. And this got Sacktor thinking, how is that even possible? Because here's the thing, the molecules in your body are constantly breaking down and being replaced. Most of them only last days, weeks, maybe a few months at most. So how can a memory last 60 years when the molecules that supposedly store it are gone in a matter of weeks? This question was actually first asked by Francis Crick back in 1984. Yeah, the same guy who helped discover DNA. He pointed out this massive biological mystery that nobody could explain. How do memories survive when everything that makes them is constantly falling apart and being rebuilt? Well, Sacktor became a neuroscientist specifically to answer this question. And after decades of research, he and his team just published something that might finally solve this puzzle. So here's what they knew going in. When you form a memory, the connections between your brain cells, called synapses, get stronger. Think of it like this. If your brain cells are cities, synapses are the roads between them. The more you use a road, the bigger and better it gets. Scientists have this saying, cells that fire together, wire together. Back in the early 90s, Sacktor was in his lab stimulating slices of rat hippocampus. That's the part of the brain that handles memories of events and places. Every single time he did this experiment, he noticed something interesting. There was this one protein that kept showing up at higher levels in the synapses. By the 4th time, he was like, this is it. The protein was called PKM zeta. Terrible name, I know, but this little molecule seemed to be the key to everything. When memories formed, PKM zeta levels went up. When he blocked PKM zeta an hour after a memory formed, the memory disappeared. It was like he had found the molecular key to long-term memory. Then, in 2006, Sacktor teamed up with Andre Fenton, and they did something kind of crazy. They trained rats to remember something, then blocked PKM zeta a day later, or even a month later, and boom, the memories were gone, just erased. This was huge news in neuroscience. Labs around the world started confirming it worked with all kinds of memories, fear memories, taste memories, you name it. But then, disaster struck. In 2013, some researchers genetically engineered mice that had 0 PKM zeta, like none at all. And guess what? These mice could still form memories just fine. Suddenly, everyone was questioning whether PKM zeta was really that important after all. Most scientists would have given up at this point, but Sacktor and Fenton knew they were onto something. They discovered that when you don't have PKM zeta, your brain uses a backup molecule instead. It's like your brain has a spare tire for memory formation, which makes sense. Memory is so crucial for survival that evolution wouldn't leave it dependent on just one molecule. But this raised a new question. If there's a backup molecule, how does it know where to go? How does it know which synapses to strengthen? It took them another decade to figure it out. The breakthrough came in 2020 when a graduate student was presenting some findings and someone in the audience suggested looking at this other protein called KIBRA. Now, KIBRA is what's called a scaffolding protein. Think of it like an anchor that holds other proteins in place inside the synapse. When they investigated, they found something amazing. PKM zeta and KIBRA were binding together. And not just temporarily, they were forming these persistent complexes that lasted way longer than either protein on its own. Here's the brilliant part. These two proteins stabilize each other by forming a bond. So when one protein breaks down and needs replacing, which happens all the time, the other one stays in place, holding the spot. It's like a relay race where the runners keep swapping out, but the baton never stops moving forward. They tested this in all kinds of ways. They made the proteins glow so they could see them binding together after synaptic activity. They gave mice drugs to prevent the proteins from binding, and the mice couldn't form new memories. They disrupted the bond 4 weeks after a memory formed, and the memory vanished. It was like he had found the molecular mainspring key to long-term memory. So here's the answer to Crick's mystery. Your memories don't depend on molecules that last forever. Instead, they depend on a partnership between molecules that keeps renewing itself. KIBRA acts like a GPS marker, showing PKM zeta exactly which synapses to strengthen. And PKM zeta keeps those synapses strong, maintaining the memory. When one partner degrades, the other one is there to guide its replacement to the exact right spot. Think about what this means. That memory Sacktor has of his sister isn't preserved by the same molecules that were there 60 years ago. It's preserved by an unbroken chain of molecular partnerships, each one passing the memory forward to the next, like a message passed down through generations. This discovery doesn't just solve a decades-old mystery, it opens up all kinds of possibilities. If we understand exactly how memories are maintained at the molecular level, maybe we can develop better treatments for conditions like Alzheimer's or PTSD. Maybe we can find ways to strengthen important memories or weaken traumatic ones. Of course, there's still debate about this. Some scientists think memory might be stored in other ways too, not just through synaptic connections. David Glanzman at UCLA points out that in some experiments with sea slugs, memories that seemed erased could actually be brought back, suggesting there might be other backup systems we don't know about yet. But even skeptics agree this is a major piece of the puzzle. After 40 years of searching, we finally have a plausible explanation for how your brain can hold on to a memory for a lifetime using molecules that only last a few weeks. What do you think? Is it mind-blowing that your childhood memories are maintained by this molecular tag team that's been passing the baton for decades? Like and subscribe if you enjoyed the video.